Welcome back to the deep dive. Today we're tackling a really interesting paper just out in Nature. It digs into a problem that, well, chemists and biologists have been wrestling with for a long time.
Yeah, how do you power these super precise enzyme reactions cleanly?
Exactly. Without all the usual mess.
And the headline here, I think the really counterintuitive bit is, what if you could just ditch the chemicals, the fussy helpers, and just use electricity? Plug it in.
Just plug it in. Seriously.
Seriously, this paper shows a pretty amazing way to do just that, specifically for making these really important pure mirror image molecules, the kind you need for drugs and things. It's potentially a game changer for green chemistry.
Okay, wow. Let's dive into this then. Our mission today, really unpack this paper. We need to figure out how they actually managed to connect electricity to, well, delicate enzymes.
Right, and what that really means for making complex molecules.
And crucially, why this matters to you, whether you're actually, building molecules in a lab or just interested in how science is making things cleaner, more efficient.
Yeah, it's got broad implications.
So let's get some background first. We know enzymes are like nature's super catalysts, right? They're amazing at speeding up very specific reactions.
Unparalleled, really. Especially when you need just one specific mirror image version of a molecule chiral synthesis, as you said.
Like needing just the left hand, not the right.
Exactly like that. They're molecular surgeons. But there's often a catch. Many enzymes, especially the ones doing oxidation, taking electrons away, they need partners, cofactors.
Like NADPH, those energy carrier molecules.
Yep, those are the classic ones. But using them, or more often recycling them so the reaction keeps going, that usually means adding other chemicals.
Which means more waste, more complexity.
Exactly. It makes it harder to scale up affordably. It's messy.
Okay, so that's one side. What of the electricity angle?
Right. So separately, chemists have been looking at electrosynthesis using electricity directly. It can be super clean. Electrons are your regent.
No leftovers.
Ideally, yeah. But the big hurdle, the real challenge, has always been getting that electricity to work smoothly with the enzymes. Biology is delicate,
Wiring up biology.
Yeah.
Sounds tricky.
It is. This paper is a big step in this field, electro-biocatalysis. How do you make that connection work?
Got it. And they specifically focused this electrical wiring effort on something called dynamic kinetic oxidation, or DKO. Remind us what DKO does.
So DKO is a pretty smart way to deal with chirality. Often you make a molecule, you get a 50-50 mix of both mirror images, that racemic mixture.
The left and right hands together.
Exactly. DKO is a process designed to take that mix and cleverly convert almost all of it into just the one version you actually want. High purity.
So you don't waste half your stuff.
Precisely. You avoid just throwing it away.
Okay, so the researchers were essentially asking,
can we power the oxidation part of that DKO cycle, not with chemicals, but directly with electricity hooked up to the enzyme system? That was the core question.
Driven by what, curiosity? Or a practical need?
Both, really. There was a fundamental gap. How do you integrate electricity and enzymes effectively for this kind of reaction? And also a very practical need for greener, better ways to make these valuable chiral molecules.
Makes sense. Solving a puzzle and a problem. So, okay, the big question. How did they actually do it? How did they wire up the enzyme? What's the setup?
Right. This is the really clever part. They built this specialized system. They call it a heterogeneous electroenzyme system.
Heterogeneous meaning?
Meaning not everything is just floating around mixed together. They took their main enzyme, an alcohol oxidase called ChOx, and they stuck it onto a solid surface.
Okay.
Specifically onto a carbon felt electrode. You can think of the carbon felt like the electrical wire where the electrons go in or out.
But the enzyme doesn't just grab electrons from the carbon directly.
Yeah, needs a helper, a go-between. So they also attached a special molecule, a rhodium complex that self-assembles right there on the electrode surface alongside the enzyme.
A rhodium complex.
Yeah.
Sounds fancy.
It is a bit, yeah. It acts like a tiny mediator, a molecular shuttle. It takes electrons from the alcohol molecule the enzyme is working on.
Okay.
And then passes those electrons over to the carbon electrode. And by applying just a small voltage, only about 0.4 volts, the electrode and this mediator basically suck the hydrogen and electrons right out of the alcohol.
So that's the oxidation step done electrically.
Exactly. They completely bypass the need for oxygen or peroxides or any chemical oxidant. The electrode system is the oxidant and pulls the electrons out.
That is really neat. The electrode and the rhodium thing acting together as the electric oxidant.
You got it. Direct electron withdrawal.
Okay, that explains the oxidation, the O in DKO.
Yeah.
But you mentioned DKO is dynamic kinetic. What about the DK part? How does the recycling the dynamic bit work here?
Right, good question. You need that recycling loop. For that, they added a second enzyme into the solution. This one, alcohol dehydrogenase, or ADH, wasn't stuck to the electrode. It was just floating around.
Okay, second enzyme, what does it do?
Well, remember the first enzyme, ChOx, on the electrode? It prefers to oxidize one mirror image, the one they want.
Right.
The ADH enzyme comes in and works on the other mirror image, the undesired one, the one that reacts slower with ChOx.
And it does what with it?
It reduces it. It basically converts it back into the starting material, back into the racemic pool.
Ah, I see. So it recycles the unwanted version.
Exactly. So you get this cycle going. The desired form gets oxidized by the electrode setup and is effectively removed. The undesired form gets grabbed by ADH and recycled back to the start.
So everything eventually gets pushed towards the desired product.
Precisely. It's like a unidirectional funnel driven by the electricity.
Yeah.
Every molecule, no matter which hand it started as, gets pushed towards the single product they want.
And that's how they get such high purity.
That's how they did it. After about 6 hours, they reported getting over 99% antiomeric excess E, which is, incredibly pure for this kind of transformation.
Wow, over 99% E. That's seriously impressive. And it sounds like a really elegant integration of two enzymes and the electrical input. Did it just work for one specific alcohol?
No, and that's important, right? For it to be useful. They tested it on, I think, 15 different secondary alcohols, and it worked pretty well across the board. Yields were generally good, mostly in the 85% to 92% range for the desired product. So it shows the system isn't just a one-trick pony, it's reasonably robust.
Versatile. Okay.
Right.
But, doing it in a small flask is one thing. Can you actually make useful amounts this way? Did they look at scaling it up?
They did, yeah. That's another key part. They showed it could work on the gram scale.
Yeah, that's more like it.
And they used a continuous flow electrolysis cell to do it. That's significant because flow systems are common in industry. It suggests this isn't just, a cool lab experiment. It might actually have legs for larger scale production.
Potential for industrial use then?
Potentially, yes. They even mentioned working with an industrial partner to see if it could go even bigger.
Interesting. And you mentioned this involved multiple universities.
Yeah, it highlights how collaborative this kind of science often is. Teams from Nanjing University and the University of Science and Technology of China. You absolutely need that mix, right?
What kind of mix?
Well, you need the electrochemistry folks who really understand electrodes, electron transfer, building the cell.
The electrical engineers almost.
Sort of, yeah. And then you need the biochemists, the life scientists, who know the enzymes, how to handle them, maybe even tweak them, and how to test them with different molecules. It's that synergy.
Combining those different worlds.
Yeah.
And what about the specific people involved? Any insights there?
Well, the senior author, Xiaoqiang Huang, apparently has a background in using light to drive enzymes' photoenzymatic stuff.
Ah, so using energy input, just a different kind.
Exactly. You can kind of see the thought process, maybe. Moving from light energy to electrical energy to power these biological catalysts. It's a smart connection.
And the first author?
Beibei Zhao. Came from an electrochemistry engineering background, so they really had the challenge of, getting this high potential electrical system to work nicely with these quite sensitive proteins without frying them.
Not a trivial task, I imagine.
Definitely not. Took real skill and probably some bold experimentation to bridge that gap successfully.
Okay, so let's pull back a bit. Zooming out, what's the really big innovation here? What's the core breakthrough?
I think the main conceptual leap is this. It seems to be the first time anyone's driven a dynamic kinetic oxidation using only electrons from an electrode as the oxidant.
No chemical oxidants involved at all in that sense.
None in the actual DKO oxidation step itself. They completely replace them with electricity. It really pushes this kind of biocatalysis into, maybe electrosynthesis 2.0, using electricity for complex, sensitive biological transformations, not just simple stuff.
And the practical advantages. We touched on waste.
Yeah, that's huge for green chemistry. No chemical oxidants means no nasty byproducts like heavy metal residues or dangerous peroxides, which you often get with older methods.
That has to be appealing for industries like pharma, right?
Yeah.
With all the regulations.
Absolutely. Cleaner processes are increasingly important. Plus, like we said, the fact that it seems compatible with existing flow reactor technology is a major practical bonus. It could potentially slot into existing production lines.
So cleaner, potentially scalable. What does this mean looking forward? What does it inspire? Where does research go from here?
Oh, it's hugely inspiring, I think. It really opens up the idea that electricity could be like a universal cofactor, a clean energy source for all sorts of different enzymes, way beyond just this DKO reaction.
So other researchers might now try to wire up
Right.
different enzymes.
Exactly. Think about all the other oxidation or reduction reactions out there. Can we hook those enzymes up to electrodes too? Could we design completely new reaction pathways this way, or just make existing ones way more sustainable?
It opens a lot of doors.
It does. Of course, there are still challenges. They point this out too. Things like making the enzyme system last longer on the electrode improving catalytic lifetime. That's always key.
And the cost. That rhodium mediator sounded expensive.
Yeah, finding cheaper, more robust electrode materials and mediators is definitely on the to-do list. Moving away from precious metals would make it much more economically attractive for large-scale use. But the key thing is, this paper provides a really strong proof of concept. It shows a viable path forward.
A clear route towards cleaner biocatalysis using electricity.
That's the takeaway. It's a validated approach now, which is really exciting for the field.
So replacing chemicals with electrons for super precise, high-purity enzyme reactions. A really fundamental shift with huge practical potential. That was definitely a deep dive into green electrobiocatalysis.
Absolutely. And it makes you wonder, doesn't it? If you can successfully plug enzymes into an electrical circuit like this, what else could you power or control electrically in chemistry or biology?
What other energy sources could we tap into?
Right. What completely new kinds of reactions could this unlock? Or how much greener could existing processes become? It really pushes you to think differently about energy and catalysis.
Definitely food for thought. Thanks for joining us for this deep dive. Keep exploring.